Good afternoon, everyone. Welcome to our coffee seminar. I'm Guangli Feng. Today, sorry, I'm Guangli Feng. I'm a postdoctoral research fellow from the Center for Animal Science. I acknowledge your traditional owners and their custodianship of the lands on which we meet. We pay our respect to their ancestors and their descendants, who continue culture and spiritual connections to country. We recognize their valuable contributions to Australian and global society. Just to let you know, today's seminar is scheduled from 12 p.m. until 1 p.m. By the end of the presentation, we will have a session of a QA session. Please enter your questions in the QA tab. You're welcome to write your questions during the presentation. We'll answer them at the end of the presentation. Today we have Viet Hai Tran, also known as Lenny. I've known Lenny for more than 3 years. Lenny is a third-year PhD candidate at the Center for Animal Science. He had a Master of Philosophy from the School of Agriculture and Food Science before starting the PhD. His research explores how the gut microbiome shapes animal health and performance, with a particular focus on post-weaning piglet. Through nutritional interventions combined with metagenomic approaches, he aimed to uncover practical strategies to modulate the gut microbiome and to support a healthier and more resilient piglet. Today, his topic is focused on modulation of gut microbiota to improve piglet gut health during post-weaning. Here you go, Lenny.
Hi everyone. Before I start, I would like to acknowledge the traditional owner and custodianship of the land that we met and pay the respect to their ancestors and their descendants. So today, I would like to talk about my work that I've done during my PhD. So we try to modulate the gut microbiota of the piglet. To address the issues that we call the post-weaning syndrome. So weaning is a stressful process for the piglet. They are separate from the moms. They were transported into the new environment, to the new stress, and they have to adapt with the new piglet, the new mate in the pens. Importantly, their diet is completely changed from the liquid milk to the solid feed. So apparently, in the first few days after weaning, they eating behavior tend to change. Some of the piglet they refuse to eat, or we call it that anorexia, and they usually has a digestion problem because of the immature gastrointestinal tract that they sometimes hasn't adapt with the change of diet. And apparently they got microbiome also disturbed. And this post-weaning syndrome cause a very negative effect on the piglet health. They slowing the growth of the piglet. They make the piglet more susceptible to the infection or the disease. And often we see the piglet had the diarrhea in the first few days after weaning. So in this in the scope of my thesis, I try to focus on to regulate the gut microbiome and to address the post-weaning syndrome. So first, I want to talk a little bit about the gut microbiome. So they are the community of the microbes that live in the gut, especially in the colon, where they harbor a very large amount of the microbes. So the activity of these microbes, they closely interact with the gut cells in many different ways. So the most important pathway is the gut microbiota. They start to utilize the the digester in the colon. So usually it's a fiber which can resist the digestion of the small intestine, and they fermented those fiber and produce such fatty acids, which serves the energy source for the gut cells. So the interaction between gut microbiota and the gut cells, they usually influence another part of the gut, which is sometimes the immune system through the famous pathway, which is cytokines, is a signaling compound. And the interaction between gut microbiota and the gut can also influence the behavior of the animal, or we can call it a gut-brain axis. As for the gut microbiota, the diverse and balanced gut microbiome, it have the properties called colonization resistant, where it can resist the infection from the pathogenic bacteria. It works closely with the immune system. So to promote a diverse, balanced gut microbiome, we in the thesis we try to formulate the post-weaning feed, the most suitable for post-weaning feed. The post-weaning feed for the piglet, they compose from three factors: the ingredient itself, the feed processing, which create a different particle size of the ingredient, and in practice, people usually add exogenous enzyme to increase the digestibility of the post-weaning feed, because as I mentioned before, the piglet, after weaning, they tend to have a digestion problem. So in the literature review, the impact of these factors which compose the post-weaning feed in the digestion were widely studied. However, the influence on the gut microbiome composition in the large intestine was not thoroughly investigated. So in my thesis, I've tried to study the fermentation rate of each factor and how it influence the gut microbiome composition in the colon. So in order to do that, first we ran a series of in vitro experiments. So the aim is to see how the microbiome composition respond to each ingredient under the influence of the particle size and enzyme treatment. So first, we study three different common grains which we use in the post-weaning feed, which is barley, wheat, and maize. We separate them into two particle size, and then we add 5 different enzymatic treatments. So control is the sample without any enzyme, phytase, protease, a cocktail of plant cell wall degrading enzyme, including the silanase, cellulase, glucanase, or we call it MOE, and mixture of all enzymes with it all. So a little bit about the procedure, how we make the digester, the dry digester. So we have 3 ingredients, 3 grains. We separate into the small or large particle size, and then we're adding an enzyme. And then we use the in vitro digestion model to digest this sample. And then we wash the soluble component, only retain the insoluble, and we freeze drying them to retrieve the dry digester. And after we get the 3 unique dry digester, this is the main part. We're starting to do the in vitro batch fermentation. So we use the medium B, which is specified for anaerobic fermentation. And for the inoculum, we use the pool of the lactation piglet who's about to be weaned in the commercial farm. So we use the pool feces as a representative of the gut microbiome composition. Then we ferment it for 48 hours. So we, after fermentation, we were able to record the cumulative gas kinetic. It will reflect the fermentation rate of each sample. We measure the short-chain fatty acids because they are a byproduct of the fermentation. And we also did the microbial profiling to see the gut microbiome composition. And first, we analyze the composition of the dry digester. As you can see, there's a difference between large and small particle in barley and wheat in case of starch content. So the large particle, they have more starch content compared to the small particle. And in case of the maize, the large particle also have the higher starch content compared to the small particle, but the degree of differences is lower compared to the barley and wheat. So after fermentation, we also found that the large particle of barley and wheat, they generate more gas compared to a small particle. In case of maize, it's also significant. The large particle of maize also significantly increase the gas production compared to the small. But again, the level of differences is lower compared to barley and wheat. So the large particle, they have more starch. They increase the gas production during fermentation. We also analyze the short chain fatty acid content, and we include in the acetate, propionate, butyrate concentration. We have total short chain fatty acid concentration and BRCHPR, which is branch chain proportion. So we analyze the branch chain acid concentration, and we calculate the proportion between branch chain acids total over the total short chain fatty acids. And then this ratio reflects the protein fermentation rate in the sample. So here you can see there's distinctly two different clusters, the large particle and small particle in case of both poultry, barley, maize, and wheat. But in case of barley and wheat, the large particle, they characterize, they tend to have more short chain fatty acids concentration. It's a little bit different in case of the maize, where the large particles, they tend to have more butyrate. So in case of the microbial composition, we use beta diversity, plotting using the PCOA to see the differences between sample. And here, the particle size is the main factor that drives the differences. So you can see there's two different clusters, the small particle and the large particle. And the difference is more in case of the barley and wheat compared to the maize. And they all derive from the initial inoculum and the blank. The blank is an inoculum fermented without any substrate form. So this is a composition of the gut microbiome in the sample. So as you can see, there is a strong dominant of the succinate Brio, which is the blue one. In case of the large particle barley and wheat, in case of maize, the succinate Brio, they dominate both large and small particle. And all the sample, the composition is entirely differently from the inoculum and the blank. So we applied the statistical analysis, and we found out that all the sample, they actually enrich the succinate Brio from the initial inoculum, but at different degree. So the large particle, they enrich more compared to small. And interestingly, in the small particle, they enrich the group of bacteria that utilize fiber as carbohydrate source, such as the Prevotella, Fibrobacter, and Lactobacillus. And then we do the Smear correlation between the short chain fatty acids and the core microbe abundance. So we found that succinate Brio and Selemonas, they actually are positively correlated with the short chain fatty acids production and the starch content as well. While other bacteria, Lactobacillus family and Prevotella genus, they are correlated with the other undigest component, which we think they compose mainly from the insoluble fiber after the digestion. So about the effector enzyme, we found the effector enzyme only in the case of the large particle of maize, where the mold and all is reduce the short-chain fatty acid production compared to the control. And more, the cocktail plant cell wall degrading enzyme also promote the group of the bacteria that utilize the non-starch polysaccharides or fiber. So in conclusion, we see that the barley wheat share similar fermentation trait, where maize fermentation is a little bit different because we found that in the large maize there was the effect of the enzyme that reduce the short-chain fatty acids and promote the growth of the fiber degrader. And then large particle, which higher starch content, actually strongly stimulate the gas and short-chain fatty production, stimulate the fermentation compared to the small particle. However, the large particle, they also strongly stimulate the succinate vibrogenous and reduce the diversity in the community. Meanwhile, in the opposite, small particle, they stimulate the bacteria more evenly, especially in the group of the bacteria that utilize the non-starch polysaccharides, or we could call it fiber. So that's the fermentation trait of the 3 different grains. And in the post-weaning diet, we also need the protein sources. So next we conduct an in vitro experiment with the 3 different protein sources. So soybean meal is the common protein sources in the using in the post-weaning feed for a piglet. And we second candidate is canola meal. Canola meal recently getting more attention because they also contain high crude protein content. And more importantly, they can be natively grown in Australia, while soybean meal is imported from overseas. And another candidate is lupin. So similarly, these 3 ingredients, each ingredient we separate into 2 different particle size and add 5 different enzymatic treatment. So a little bit difference is in this experiment, instead of using the cocktail of 3 different plant cell wall degrading enzyme, in this experiment we only use glucanase. So for the composition of the dry digester, there's almost no starch content except some rare case in canola meal, large particle of the canola meal. And the protein content between large and small particle size of canola meal is basically very similar. The protein content in case of soybean meal a little bit reduced between large and small, and highly reduced in case of the lupin between large and small particles. And after fermentation, we found that the large particle canola meal actually have the lowest gas production compared to soybean meal and lupin. And even small particle canola meal also have a lower gas production compared to soybean meal and lupin. So the canola in general has the lowest gas volume compared to others. So it means that the canola is harder to be fermented. So the results from short-chain fatty acids also shows the fermentation trait. Case of the soybean meal and lupin, we can still see the effect of the particle size because there's 2 clusters separately, but we couldn't see the clear cluster between large and small particles in case of canola meal. And the statistical analysis shows that the large particles of soybean meal and lupin promote the production of butyrate compared to the small one. So in case of the enzyme effect, interestingly, we found that the phytase actually promotes such a fatty acid production compared to control. It's not protease, it's not the glucanase, but it's the phytase. And in case of the microbial composition, in the protein source, the ingredient itself is the main source actually drive the difference between the sample. It's not a particle cell anymore. And actually we can still see the difference between large and small particle size in terms of the lupin. And the statistical analysis show us that most of the sample in which the bacteria belong to the group of the fiber degrader, such as the Lactobacillus, Fibrobacter, or Prevotella. And in case of the lupin, they actually also enrich succin in vitro compared to the initial inoculum. So we also analyze the predictive function in genomes, and we found out that the gene and function that related to the plant cell degrading, such as the beta glucosidase and beta galactosidase in the group of carbohydrate metabolism, were also enriched in all the samples. And the Spearman correlation actually shows us very interesting results. So it shows us the genera belong to the Lactobacillus family. They both positively correlated with the butyric concentration and the protein content in the ingredient. So in the literature, the high crude protein content in the diet in the post-weaning diet often stimulate the protein fermentation in the hingut, in the colon, and it sometimes it creates a byproduct such as ammonia, which is highly toxic for the gut cells, and sometimes it also proliferate pathogenic bacteria. But here we identify the group of bacteria that both positively correlated with such hepatitis production and the protein content. So the enzyme effect is also differently in soybean meal, canola meal, and lupin. In soybean meal lupin, the enzyme effect highly depend on the particle size, but not canola meal. So the enzyme effect on the canola meal is independent from the particle size. So in conclusion, all the sample, all the protein source stimulate the non-start polysaccharide degrading bacteria, but at different degree. Canola meal generates less gas, less such fatty acid production, and it's more difficult to be fermented compared to soybean meal or lupin. We can see the particle size effect in case of soybean meal and lupin, but not in canola. Interestingly, the combination between phytase and large soybean meal actually promote such fatty acids production. And we were able to identify the potential benefit of some genera in Lactobacillus family in the gut fermentation. So the in vitro fermentation show us the fermentation tray of different ingredients, how particle size influence the fermentation of each ingredient, and the role of the enzyme treatment as well. So next step, we want to see if the result is applicable in the practice. So we conduct the animal trial. So in the animal trial, we use the wheat barley as the main protein sources because they have similar fermentation trait. And we in the animal trial, we want to focus more on the differences between the soybean meal and canola meal and how they interact with enzyme. So we conduct our animal trial with a unique post-weaning diet made from 2x4 factorial design. So a diet made from two different protein sources, which is soybean meal, either or canola meal, and we mix with for another 4 enzyme treatments: control, protease, move, which is a cocktail of plant cell wall degrading enzyme, and all, which is a mixture of all enzymes. And here we add phytase in all the dietary treatment. So for the animal, the 72 piglets were directly weaned from the commercial farm to our research facility. And then we feed the piglet ad libitum with our dietary treatment. On the day 9, we sampling the sample in the large intestine, where we sample from the cecum, proximal, middle, and distal colon. And first, we didn't find any significant difference in terms of the growth performance, such as the weight gain, average daily weight gain, or feed intake. However, we find a significant difference in terms of cecum weight, which is the net weight of the cecum without any digester. So the protease group actually have larger cecum weight compared to the O, which is mixture of all enzyme. And we also found the differences in terms of diarrhea rates, which reflect the rate of diarrhea incident in between the groups. So the protease group, they have lower diarrhea compared to the move. And we analyzed the short-chain fatty acids from the cecum, middle colon, and distal colon. And using a statistical analysis, the model shows us that the dietary treatment influence the short-chain fatty acids independently from the gut section because there's no interaction between them. So, which means the dietary treatment influence the colon, the different section in the colon at the same degree. So here in the ANOVA table, you can see most of the differences was come from the gut section factor. So in detail, in case of a short-chain fatty acids concentration, the cecum generate more short-chain fatty acids compared to middle colon and distal colon. And interestingly, in terms of enzyme treatment, the protease, they actually generate less lactase concentration compared to all across the colon. And in terms of short-chain fatty acid yield, which is the total short-chain fatty acids, the middle colon is the section that has highest short-chain fatty acid yield. And in terms of protein sources, soybean meal is yield more short-chain fatty acids compared to canola meal across the colon. And interestingly, in the middle colon, we also found that the soybean meal generates more short-chain fatty acids concentration compared to canola meal. So these results actually align with the in vitro fermentation, where soybean meal was found to have higher short-chain fatty acids compared to canola meal during fermentation. So we also conduct the spiral correlation in each section. So in colon, the short-chain fatty acid that generate in colon are highly correlated with the cecum weight and the cecum weight proportion over the body. So in cecum, the short-chain fatty acids is correlated with the weight of the cecum. And actually, the propionate concentration is the one that correlated with the weight of the cecum. It's a little bit different in terms of the middle colon. So in the middle colon, the butyrate concentration is generated in the middle colon, correlated with the weight of the middle colon and the weight proportion. And there's almost no correlation in terms of the distal colon. So to analyze the microbial composition in this animal trial, we used metagenomics analysis. So we used the Oxford nanopore sequencing to sequencing the DNA extracted from sample that we collected from the cecum and the distal colon. For the metagenomic analysis, we use two different database for taxonomic classification. The GTDB, which focus on the bacteria and the archaea, and NCBY, which also include the eukaryotes and virus. For the functional binding, we use the SEDD database and KEEG database. So as you can see in the beta diversity, the composition of the microbial composition at species level in the sample, they are not they're highly similar because there's no clear cluster. There's actually two clusters that are related to the gut section, which is a cecum and distal colon, but they still highly overlapping. And this is a core microbiome at genus level using the GTDB database for taxonomic classification. As you can see, there's also the most prevalent and common genus is Prevotella. And then we can also see the Succinate vibrio is also prevalent among the sample. So they seem to respond to the baleen with in the post-weaning diet. So the statistical analysis show us the impact of the enzyme on the particular bacteria. So here we can see the proteins, they actually have more impact compared to the mold and all because they influence more bacteria in the community. So they tend to enrich the bacteria that can utilize the carbohydrate to produce the short-chain fatty acids. And here we can see the association of the bacteria that were influenced by the interaction between protein sources and enzyme treatment. So, for example, in terms of canola meal, the mixture of all enzyme is significantly stimulates the Bacillus monas, which is a novel species associated with healthy gut, but it's also significantly decrease the FDR GABA virus. So this species actually consume GABA. GABA is a compound that's linked directly to the gut-brain axis. They still under study, and scientists found this component highly regulate the mood of the animal, and some call them the common agent. So the reduction of this species is at the moment it's not clear. Is it beneficial or is it have a negative negative effect? But this is evidence that the enzyme treatment that can directly influence the gut-brain axis. So in terms of soybean meal, proteins still have more impact compared to the mold and all because it tend to enrich the healthy gut microbiome, the healthy microbes. So functional annotation based on seed database also reveal interesting results. So the mixture of all enzyme actually promotes the virulent factor ESA6. So ESA6 was originally found in the pathogenic bacteria, where it's create the protein that can rupture the host cells, and also enrich the antimicrobial resistant functions as well. So apart from the bacteria, we also use NCBY database to do the taxonomic classification. And apart from the famous bacteria, fiber degraders, Secatella, Prevotella, we actually found a high abundance of a family called Viricetes, the green one. So this is a bacteriophage, and they are fairly prevalent, especially in the baseline sample, which is a sample when the piglet just arrived at the weaning facility. And the statistical analysis also show us there is interesting result in case of the soybean meal where the mixture of all enzymes significantly enrich the abundance of the single cell eukaryote, which belong to the cilioflora. So they are the common environment single eukaryote cells that usually found in the wet area. We actually look back at the procedure and the data and the data filtering to make sure this is not contamination. And at the moment, we haven't had the explanation for this enrichment, but this is evidence that shows apart from the bacteria, the enzyme treatment can also influence another microbe as well. So for the functional annotation using the KEGG database, we actually found out that the soybean meal significantly enrich inositophosphate metabolism. So this pathway are actually related to the phytate degradation in the sample. And the significant enrichment in soybean meal group compared to canola meals is showing that the phytase. So in all the our dietary treatment, we add the phytase, right? So the combination between the soybean meal and phytase actually promote the phytate degradation in sample, and this actually can explain the promotion of such fatty acid production in the in vitro fermentation between soybean meal and the phytase. And in terms of the enzyme treatment, in the soybean meal, the mixture of all enzymes also promote the pathway related into phytate degradation. So, with or without the enzyme, is also stimulate the phytate degradation in sample. So for the conclusion, the impact of dietary treatment are the same across the closed session because there are no interaction between the dietary treatment and the gut session. And this animal trial also also confirm the result that the soybean meal generate more short-chain fatty acids compared to the canola meal. And the combination between soybean meal and phytase actually um, enrich the phytate degradation pathway, which might explain the stimulation of the short-chain fatty acids of this combination during the in vitro fermentation. And based on the results from the physiology and from the gut microbiome composition and functional of the microbiome, we think that especially for the piglet, newly weaned piglet in the first week, using the protease separately at the individual enzyme might have better benefit compared to the mixture of all enzymes. Because in the literature review, most of the time to maximize the digestibility of the feed, people tend to use the cocktail enzyme or mixture enzyme. But in terms of the gut microbiome and their function, maybe we suggest that proteins are separate individually. Enzyme may have provide better benefit. And we found out the mixture and all enzyme, they might enrich some potential virulent factor. So here's a take-home message. So my PhD focused on the investigation of the influence of the diet on the gut microbiome, especially on a newly weaned piglet. So the intro fermentation shows us the fermentation trait of each ingredient and how the particle size influence this fermentation. And we also look at the enzyme, the effect of the enzyme treatment as well. And in the in vivo experiment, the result from the animal trial help us to explain some of the result in the in vitro experiment. And we also confirm that the soybean meal, they still generate more such fatty acids than canola, and all in all, we hope that this species work would provide a more comprehensive method and procedure to actually, when we talk about the investigate the way to modulate the microbiome, especially in the piglet. So that's the end of my presentation. I would like to thank my my supervisory team and my colleague and research officers. Without them, I can say the work that I have done would be nearly impossible. And I also want to acknowledge industrial partner for the help. Thank you.
Thanks, Lenny. Very interesting talk. I really like the approach of the combination of in vitro experiment and also in vivo. Using the in vitro experiment, you can select the ingredient, like you can put into the in vivo experiment, which would may reduce the animal use. We have two questions in the QA. So the first one is for protein ingredients. Why did you not use cellulose, in particular with canola meal?
So when we designed the experiment, we actually have a discussion with our industrial partner who actually produce enzyme, and from the unpublished data, they suggest that for the soybean meal, canola meal, the glucanase may have better effect. So that's why we use the glucanase only. And yes.
I think the question was about cellulose, not the
Yep.
enzyme too.
No, because in case of in vitro fermentation, we use the glucanase only, so the VP. Yeah.
The next question, do you think the protease could digest other enzymes? I've noticed this as well because for protease, in your animal trial, you said it has high CK content, compared to the all group. As your all group, it would contain all the enzyme. So it seems it works less effectively than the protease by itself. So do you think this was a problem with the all combination in comparison with the protease itself? Does the protease could digest other enzymes?
It possibly, but again, in the scope of my experiment, I didn't measure the enzyme activity in the digester, so I actually not sure is it happen or not. But is it one of possibility that can happen that can answer the result that we have? A really good question.
Yes, no more question at the moment in the QA. While we're waiting for other people to write their question, I have a question I want to ask. For your in vitro experiments, it looks like particle size has a major impact than enzyme treatment. However, for the animal trial, you focus on enzyme treatment and the two protein source. But you sort of ignored the particle size effect. Why is that?
So for the in vitro fermentation, especially for the grain, we found out that the large particle size, although they generate more such fatty acids and gas, but they actually reduce the diversity of the community, because there's a domination of the 16 in vitro. While the small particle, they generate less such fatty acids, but they promote more diverse microbial composition in the sample. So that's why we think that to have both, like to have high such fatty acids produced and high diversity of the microbiome, the mixture between both of them would be better. So that's why in the animal trial, we provide the feed at the in the mass form where the large and small particle is mixed together. So it may enhance the actually enhance the benefit for the gut microbiome.
Mm. Um, I also have another question. In your in the first section of any in vitro experiments of the grains, so you said so probably this because with large particle size it has more starch. So I think a starch is probably the driven of the production of high production of short-chain fatty acid. But it seems for the short-term fatty acid profiles, so individually they are quite different. Do you think is it because of the different structure of starch or because of the non-starch polysaccharide in the grains?
Uh, Actually, the starch is main force to stimulate the certain fatty acids. You mean the difference in case of the maize, right?
Mm. Yeah,
So
you said maize is different.
yes,
But it seems wheat, you said, stimulates the production of butyrate.
uh, uh, yes, the starch, the starch structure definitely a little bit different between maize and so the starch structure in barley and wheat they are a little bit similar, but in
Mm.
maize it is the three structure and it's a little bit different, and the starch in the maize in the kernel of the maize they locate in different location as well. So it's
Mm.
more attached to the plant cell wall. And not to mention the uh, ratio between amylo and amylopeptides within the starch as well.
Mm.
So I think that's one of the that may be one of the reasons that drives the differences in terms of starch content between barley and maize and wheat and maize.
Mm. Thanks,
But
Lenny.
yeah.
Yeah, there's another question came in from Xianyi Liu. He said, nice talk. From the result, can we suggest that phytes could promote gut health through providing in situ as a nutrient for microbiota?
Uh, From the results, yes, we can suggest that, but unfortunately, we didn't actually measure or find a way to prove this. But from the result that we have from both in vitro and in vivo, yes, we can suggest this pathway.
Thanks, Lenny. It looks like we don't have more questions from the audience. Can we go to the next page? Lenny, could you move to the next slide? Thanks everyone for listening to today's COVID seminar. We have the next seminar is on the 7th of July. We will have Anna Kotno. Her topic would be achievement of the high gain project 2020 to 2026. Next slide. If you haven't signed up the COVID seminar, please scan the QR code or sign up of coffee.uq.edu.au. Thanks everyone. I'll see you in 2 weeks time.
Thank you.